today we are having a number of students online, and besides that, I think more than 100 students are sitting in the classrooms. They are also listening to you, Dr. Marina. They will be listening to you, your lecture, very interesting one. So before we proceed, I would like to have a glimpse of this particular program that has been sponsored by the Department of Science and Technology, that is our Ministry of Science and Technology, Government of India. And this program is open to all the scientists below the age of 40 years. And this particular program is intended to provide an exposure to the young faculty members and scientists who are working in India. And it aims to impart high-end research training in frontier areas of science and technology, which are of interest to India by providing an opportunity to visit leading institutions or universities across the globe for 2 to 6 months. The applicant who apply it, the basic qualification for them is PhD in the respective area, and the age limit should not be more than 40 years. And the SERB under this program, the SERB is the actually board of Government of India under DST, that is the Science and Engineering Research Board, that gives SIRE fellowship. SIRE is basically the short form of this, SERB SIRE, that is the international research experience for 2 to 6 months. This facility is given by the Government of India to our scientists, and they support the travel, they support miscellaneous charges, they support medical insurance, and they also give travel fare. Also, they give away $3,000 per month scholarship to the selected ones. So, Dr. Marina, I would like to introduce you with this small thing that our honorable founder president is a great visionary, and we always motivate our scientists and faculty members. And in this particular program, with the great motivation and encouragement of our honorable founder president, sir, Dr. Ashok Kumar Chauhan, 73 eligible faculty of Amity University applied for this SERB SIRE fellowship in Feb 2022, out of which 10 were selected by the Government of India, and Dr. Manish is one of them. Manish is one of the excellent faculty and scientists of Amity Institute of Biotechnology, Amity University, Madhya Pradesh, who joined us in 2015 after completing his PhD from RD University, Jabalpur. So this is in short about the program and Dr. Manish. So before we proceed, I would like to have a brief glimpse of Dr. Marina Keluzana, who is a great scientist and faculty in HDSU, that is in California. Dr. Marina Keluzana graduated with honors from Olis Honsar Dnipro National University, Department of Microbiology, Ukraine in 1994. She earned her PhD in microbiology in 2000 from Russian Academy of Sciences for Microbiology and Biotechnology, Institute of Biochemistry and Physiology of Microorganisms. Since 2001, Dr. Keluzana has lived and worked in the United States, where she is currently a professor of microbiology and lead of the C1 Biocatalysis Laboratory in the Department of Biology at San Diego State University. Her research interest includes microbial genomics and physiology, system biology, metabolic engineering of microbes that consume single carbon compounds such as methane, methanol, formate, and methylamine. Basically, her research interest includes 4 areas, which are fundamentals of C1 biocatalysis, C1 biotechnology, methane mitigation, and soil C1 microbiomes. She is the author of more than 100 scientific publications, book chapters, and many patents related to microbial methane conversion. Dr. Marina's research credentials in terms of H-index, ITN index, and total citations are 46, 95, and 7,152 precisely. I would like to request Dr. Marina for today's lecture.
Thank you. Thank you so much for a wonderful introduction. And of course, it is my great pleasure to host Dr. Manish Kumar in my lab. It was incredibly great pleasure so far, and I hope we will be able to establish collaborations kind of lasting for years and for life. Let me to start from start.
If Lieutenant General V.K. Sharma permits, and before Marina starts, I would like to say a few words why we are here today. Vice Chancellor V.K. Sharma ji, I need your permission to.
Yeah, please go ahead. Please go ahead, Professor Chakravarti.
Yeah. Marina, I think it has already been told your credentials, your background, and your excellent research work. And that we have also been told, why we started, why we sent Manish to you. Manish himself is a very extraordinary researcher, young, and with a lot of dream and aspirations in his eyes to do something for the country and also for the Amity University. Few facts I must tell you so you will get it perspective. In India, we have about 1,100 universities. About 400 plus universities are private, but the remaining are government owned. And Amity, which is a private university, it came into existence in 2005 under a great vision of one gentleman called Dr. Ashok K. Chauhan, who is the founder president of this initiative. He was in Germany for about 3 decades before he came to India with a intention that he must do something for the country. So he chose national development through education as his mantra to proceed. And that's why he created this entire chain of Amity universities. It is 11 universities in this country, plus 12 campuses outside India. Amity campuses are there in Singapore, in Dubai, in New York, in Tashkent, in Mauritius, in Singapore, and other places. So I can say it's a global university. Now, when we say a global university, it also has a lot of expectation and aspirations from its faculty, like Manish Kumar. And this scheme, under which Manish Kumar is presently spending time with you, trying to understand the latest techniques and latest research, trying to benchmark his research work with the global research work, especially on this important area of the methane mitigation through crop productions. Now, while we are all eager to listen to you, but I must put a perspective here, why we want you to be here, why Manish to be here, why we want Lieutenant General V.K. Sharma to be here. We are looking for our association, our partnership between you and Manish beyond this coming few months. Can we think?
Definitely was. Yes, he definitely was incredibly successful in establishing it. So it was a great pleasure to have him in San Diego.
So this platform
Manish.
which we have created today, and we are going to listen to you, is actually how we keep our connection strong, how Manish and other Manishes, how we can clone few more Manish and send them to you and welcome you and your team to Amity so that we can look for a great greener future of tomorrow.
Thank
Through
you.
research. Research for national development, research for the development of the world is the buzzword, and we are looking forward to listening to you and your views, how we can strengthen our connect for the future. That is the intention, and that's why we thought of requesting Manish, and through Manish to you to have a discussion. That's why we call it an interactive talk. It is not only a scientific talk only, but we are also trying to look for the possible opportunities to connect to each other. So thank you, Marina, once again. And thank you, Manish, to have it. But we would expect, if tomorrow we would like to request Marina to receive a half a dozen Manish in her lab, I'm pretty sure he will immediately agree to it. And that kind of impression you are supposed to create. So you have to prove yourself, you have to hard work, maximum, and make your impression, lasting impressions in the minds of the mentors like Dr. Marina. So Marina, once again, welcome you. I will not talk much further. And you can go ahead with your presentation. Thank you.
Thank you. Thank you. Okay, so let me just share again. All right, so again, I just would like to thank you for Dr. Manish Kumar visiting my lab, and thank you for this invitation. And of course, it is a great pleasure for me to talk about my research. And I try to shape it today in a such ways that it's kind of related to what Dr. Manish Kumar also did in my lab. And I know that he will present right after me. So I'm going to give an overview a little bit broader, and it's mostly about single carbon compounds from nature to C1 Biotech to give you a sense what we are doing and what Dr. Manish Kumar was able to explore one way or another. So what's happening here? My outline today has three parts: inspiration, knowledge, and implementation. And I come from an area, single carbon, and I know that it's something that not everybody knows immediately. So I already see that that you have a wonderful colleagues with quite nice knowledge in methane, but on a global scale, but I still would like to introduce single carbon and biosphere. And I will briefly talk about Earth's carbon as well as the Earth's climate. I want to tell you a little bit more about C1 microbes, and with a focus on arid ecosystems. I'm trying to dedicate most time for it because this is where I believe our connections might be incredibly strong. And at the same time, I will give you examples of C1 Biotech because my team really works in that area a lot. We do metabolic engineering, we modify microbes for different areas of biotechnologies. And this is another part, depending on the time, I might give you more or less details, but I definitely give you a sense what we are doing here. So let's me to start with the single carbon compounds, and we're going to talk only about organic compounds today, such as methane, methanol. It's also formate methylated amine, methyl sulfides, and many other things. But the main core is that they do not have carbon-carbon bonds. And as I present those, I also want to emphasize that when I think about single carbon compounds, I think about molecules that are energy rich. What that means? That means when microbe eats methane or methanol, it's not only source of carbon, it is also source of energy. So it's all what microbe is needed on top of a few minerals. And this is what I want you to remember as I present today. Before we start, I really like to quiz people a little bit and just let's think about Earth as a planet and think about Earth's organic carbon. I'm giving you this pie with 2%, 1%, 5%. And those percentages is associated with marine biota, land biota, peat soil, methane, and fossil fuel. And I just will give you 15 seconds to think about where do you think each of this category is? Just like very few, let's say, what you think would be the most, where is the most carbon on this planet? Is it in marine biota, land biota? Is it in soil? Is it fossil fuel? And I'm just like to do that because very often, at least when I present it here in US, it's really shapes when people really see the real numbers. When we think about carbon, when we think about Earth, it's our vision. We have ocean, it's big and everything is there, but it's really not. The marine biota represents a very small portion of overall organic carbon on this planet. Significant portion is in fossil fuel. It does not mean that we have to use it without restrictions, but it is there. It was created but a million of years. But I also want to point out that when we think about methane, it is almost a quarter of all organic carbon available on Earth. It's a lot. You will think about it's like one of the main molecules. It's the most abundant compound. It comes from fossil as well. It comes from geothermal and biological origin. And why it is critical? Because methane is an energy-rich and carbon-rich molecules. Delivers everything for life, and this life can exist without light. And this is what, as I envision arid ecosystem, I will also bring you to this point. Just think about that methane can provide energy and can provide resources on its own. And of course, we know that it plays a key role in global climate. And this is another picture which I also like to show, which was from Paris Congress, and it's more discussed right now. We know that methane is a powerful gas, but how powerful it is? It's contribute almost 45% to global warming. So I like to present it here because if you will look on CO2 and methane, they're almost equals. 45% comes from methane. Even so, the concentrations of methane are much smaller. If you look where this methane comes from, it comes from energy recovery, natural gas production, as well oil industry production. It's a little bit from aviation, road distribution of natural gas. But one of the most significant part, and this is what I also want to bring up, that will come from animal husbandry. It will come from agriculture, from biomass burning, from waste and landfills. And why this is critical? Because all this methane is biological methane. All this methane comes from the part of the cycle which produces it. And we as humans disturb this part. And in my opinion, we have to restore balance. We also have to do it in a natural way. Bring biology to complement what was disturbed before. With that, I want to point out as well that we have a methane pledge. It came out early this year and it's very promoted in US. We like to say 30 by 2030. What that basically means that US pledged to cut methane emission by 30% just in 10 years from now. But this pledge was supported by European Union and more than 100 countries around the globe. But how we can do it? Of course, there are different ways. We can reduce flaring, we can reduce venting, we can improve our infrastructure to tighten nuts and bolts to make sure that methane does not escape from natural pipelines. We can develop several approaches to mitigate methane emissions, specifically methane emission associated with livestock and agriculture. This is where it's critical and important. Now, before I will move, I believe the one key point, we know that methane consuming bacteria, and I mentioned it a couple of times, they're everywhere in nature. But does biology matter? What's exactly microbes do? And I'm going to place this small methane, and if I will go over literature and we'll try to pull together numbers. We know that fossil geothermal methane and methane hydrate have an incredibly large depository from 25 to 5,000 gigatons, a huge number of methane. And we know that some part of it from 2.5 to 5 gigaton is emitted every year. It also comes significant portion, we know that 110, 150 gigaton, this is our primary production. This is what feeds by all photosynthetic organisms on a planet, comes to biomass. And from this biomass, about 5 gigaton also will be degraded by methanogenesis, leading to methane. However, from this methane, only 0.5 gigaton is emitted to atmosphere each year. From this point, what happens with methane in atmosphere? In methane, significant portion of it would be decomposed in troposphere, in stratosphere, and small part will be sucked back, respired back by soils primarily. Now, if you will look on those, again, in anaerobic part, methane will be utilized. And it's about, if you will just balance everything, from 7 to 10 gigaton per year, microbes, which we know is methane consuming bacteria, they will take methane and convert it back to CO2. And there are different ways how microbes do it. They couple reactions with oxygen, sulfate, nitrate, metals, with many different parts. But the key point to remember that nature already has a capacity to recapture a lot. It's much bigger numbers that we like to refer to stratosphere. We just have to use and understand and utilize this capacity because it's much more than we currently have in the environment. What we also know about that we know when we think about geothermal methane, this is a whole ecosystem which is supported by microbes which can utilize methane. We can trace methane through a food chain, even in freshwater system. And I've given this wonderful study which demonstrates that about 0.8% of fatty acid in freshwater fish will come from methane. Now, let's look on the methane cycle in a global scale. We know about methanogenesis. This is how methane is produced, and methanotrophy and biological methane cycle, how methane is consumed. Both functions are widespread. Methanogenesis is mostly anaerobic, and methanotrophy, it can be aerobic, anaerobic, and I like to call somewhere between because microbes are incredibly flexible. What we did with my colleagues here with Edwards at San Diego State University, we asked a very simple question. We generated huge amount of metagenomic information about all possible ecosystems on the globe. And the one simple question we ask, where are microbes involved in methane production, and where are microbes which can be involved in methane consumption? So we run all possible metagenomes. It's about 100 terabytes of information, and results came back with about 9,000 metagenomes, 40% of everything that ever was deposited to in CBI, with about sequences ranging from 1 to 300 sequences for each 100,000 reads. And if we will map them and look where in natural ecosystem we have methane production and methane consumption. We were focusing on MCR as a way to produce methane and PMO as aerobic methane utilization. What we found, and this is again very important to what we do and what Dr. Manish Kumar did in my lab as well, that methanotrophy found primarily in terrestrial ecosystems. Soils are the main players. At the same time, if you look where methanogenesis happens, that will prevail in human-made ecosystem. It would be our wastewater, anaerobic digester, everything what we disturb. Everywhere we will have methanogenesis coming up. With this, I kind of coming to my first conclusions that natural methanotrophs reduce methane emissions significantly. They remove almost 95% of the cycle overall. Terrestrial methanotrophs are the main player, and human-made ecosystem shifts the balance to methanogenesis. And with that, how we can rebuild our ecosystem to bring methanotrophy back, to restore the balance. And I really like to split it in 3 different categories, and all of them one way or another way addressed in my lab. I talk about, we do capturing, and I will show only one slide related to that. We do a lot of conversions, and again, this is where metabolic engineering, this is where biotech came along. And we really started and explore and looking forward to explore even more how we can preserve agriculture. And this is topic which I love a lot, and this is what I was very happy to host Dr. Manish Kumar, because this is his participation also helped to explore it more in my lab. But I will give you a little bit introduction what we did before that. I would love to start with arid and semi-arid ecosystems, because this is where we live in Southern California, but it's also third of the planet biomes. We know that agriculture as well as the climate change contribute to expansion of arid and semi-arid ecosystem around the globe. And one question which when we started the project was, do micro and microbiomes have time to adapt to those changes as we transition from to arid and semi-arid ecosystem? What changes happens? And why it is important, because arid ecosystem on another side, on the positive side, are quite unique. Some of the most significant things of methane. The rates of methane consumption in arid ecosystem can run to 435 milligrams per cubical meter per day. That's very large consumption rates. And what we also know that those mostly happening at aerobic conditions. We also know it very few, very preliminary studies were done when we started to look into arid ecosystems, that the methane cycle is governed by a novel yet uncultivated microbial clade. So, well, basically, we know that it's significant thing. We know that methanotrophs are key players. What is interesting, when semi-arid ecosystems are converted to agriculture, they lose the capacity to assimilate methane, and they actually becoming a source of methane emissions. So for us, it was very important to understand what might be happening and how we can preserve it. So our model ecosystem is Anza Borrego Desert, which is like next door from San Diego. It is West County, East County, Eastern San Diego County. It has wonderful history, sedimentation from time, marine and terrestrial environmental. It passed through years, becoming from almost wetland to desert. As I mentioned before, it's desert, it gets very little water, and it has very significant methane flux. What even more important, and this is what slide I really like to show, in spring it is very beautiful. It's just, you'll love to see the flowers in a desert. But if you will look, this is Anza Borrega, and this is Salton Sea, and all these triangles show vents, methane vents. All those triangles contributes to methane emissions. However, if we will look on distribution of methane around the area, and I kind of zoom in and it, here is the San Diego when we have significant amount of methane emitted. But if we will look on this preserved area of Anza Borrega, it's blue. This is where methane is consumed. Despite that it's next door to significant emissions of methane, the whole ecosystem has a capacity to suck it in. And we really wanted to explore it more. So what we did to start, our goal was to understand mechanisms that sustain methane cycle in the desert. And we use a multi-scale approach. We measured methane in the desert. We collected samples from different depths. We did metagenomic, metatranscriptomic. We did enrichments. We also look on the structure of the community by compiling all different data sets. And I'm going to present many of you today. So number one, what was one big surprise studies which were not done before? We noticed that methane consumption in desert is linked with vegetation. And this is wonderful plots collected by my graduate student, Nati. When we have plants, even if it's like almost dead bush, it still will consume more methane just soil alone. We notice that methane consumption very often associated with many different plants in the desert. The most important to have vegetation, and this vegetation will consume methane. The next part, we looked on the metagenomic. And again, we have a huge data set, about 3.3 gigabytes per data set. We have 18 metagenomes. From these metagenomes, we identify about 520 or 16 as RNA, diversity of the community for data set. We have about 60 genomes, which we were able to assemble, those represented by 24 bacterial phyla. We were able to reconstruct networks and see interactions between different microbes, identifying positive as well as a negative. What was sticking out for us immediately is that we see these positive correlations between methanotrophic bacteria and brown rhozobialis. We also were able to assemble genomes directly from metagenomic studies, which belong to Metallocaldum. We detect a little bit Metallobacter luteus. And again, the distribution correlated with vegetation. We did a number of enrichment studies. We isolated 5 pure culture of Metallocaldum, some of them related to Metallocaldum, which we assembled in metagenome. We also isolated 8 satellite culture, which reinforce the observation on metagenomic data sets, and there is a correlation between different groups. And those satellite cultures were Bradyrhizobium and Neorhizobium. And many of you might know that those wonderful, very well-established plant symbionts. And I'm just showing you pictures. Will be Metallocaldum, sitting next to it will be Bradyrhizobium. And we also notice that they are like Romeo and Juliet. They love each other. They like to grow next to it. It took time to separate those cultures, but they have this tendency to grow better with microbes are near each other. And we explore this further. So we immediately combining that methane cycle is linked with vegetation, and there is a cooperation between 2 microbial species. We wanted to see how methanotrophic bacteria and plants interact, what might drive those interactions. Because everything started with desert, we also wanted to follow up with drought. So our plant became Bucharada papriata. It's a very simple why this plant was chosen, because it's native to North America. It's very closely related to Arabidopsis. In our microbes, were Metallocaldum and Bradyrhizobium, we established co-culture, and we did different set of analysis. We look on metabolomics, on gene expression, on plant chlorophyll content as well. We looked on how carbon as well as the hydrogen in form of a deuterium transfer from microbes also way to plants. And I'm going to present you those data. But number one is very simple experiment. And some of those experiments Dr. Manish Kumar will show for other different microbes today. But what we did, we basically took plants, we inoculated them with 2 microbes in soils which were sterilized, and we did in water plants. And we looked on what would be impact, and we comparing plants which were supplemented with microbes and were not. And without water, and I believe you don't need to see more, GMO, you can immediately from the beginning say we have the same distribution, nice healthy plants which looks like somewhat smaller. But after 14 days without watering, what was surprising to see that plants which we supplemented with metanotrophic bacteria still behave incredibly well when our control group was completely dead. Now, we looked on what might be happening, what exactly molecules are transferred between microbes and plants. And we came a little bit more creative how we can trace gas, because gas is an environment, how we can trap the interactions. So we did see for 13C methane and CD4, and we look on the incorporation. We also look on plants, and those like control group was just air with methane, with C13 methane, and with CD4. One part, every time when we add methane, even if we add microbes or not, we will see that root development will change, and we will have more roots formed for plants. Another part, what was incredibly to find that when we look on C13 incorporation, we can see flow of carbon all the way from microbes to the root. And it's not only carbon, because you can imagine that methane is oxidized to CO2 and plants fix CO2, so that will be very easy CO2 incorporation. But we also see hydrogen, which will travel as well, indicating that there is a transfer of organic molecules from microbes to plants. And one best way of looking on those was also C13 NMR analysis. When we looked on microbes, it is NMR, and then we look on plant, and we also root as well as the plant leaves. And this part of the compounds, which was all the way transferred from microbes, those compounds were present in microbes, and we can detect them in roots in plants. So we did additional study, which was metabolomic, to investigate what exactly might be exchanged between, more precisely, what can be exchanged between plant and microbes. And we found about 20 metabolites. When we compare plant and plant which incubated with metanotrophic bacteria, we can see that plant will produce certain compounds. It will be fructose, glucose, and some amino acids. Those will be accumulated in sediment, which we use a very simple sand, purified sand for metabolomic studies. However, when we compare, and most likely those will be eliminated when microbes are around. However, we compare plants to supplemented with metanotrophic bacteria versus just metanotrophic bacteria, what microbes can produce on their own. The significant portion of compounds which will be secreted by microbes, and this was a surprise to us. In addition to glycine, betaine, octane, which we expected compounds as osmolalite, which will support drought, in plants, we also found quite a few of phospholipid. It was choline, choline phosphate, as well as phosphoethanolamine. So this exchange in lipids is something new, and we love to investigate it further. And moving to kind of conclusion about arid land, we've given you some take-home messages. Desert plants and methane-consuming bacteria integrate their metabolism to function in halobionts, so they work together. Plant can transport, we predict that they will transport methane to support active methane oxidation and metanotrophic communities, building those up and building microbes which can provide additional molecules to support growth of plants. And those interactions are becoming incredibly beneficial for drought stress. And of course, you can ask me, and what, how that might be related to agriculture. And this is wonderful study which was carried out again by graduate student Nancy Delhabre, which took microbes which isolated from desert, and this time she took sorghum as a agricultural crop, and she just supplemented this agricultural crop with metallocaldum species and look on the impacts. Again, like we observe it with native desert plants, we see benefits to plant from supplementation of metanotrophic bacteria. It's not only will grow better with very low water supplementation, it will preserve photosynthetic capacity, will look incredibly healthy. It will have root structure very well developed, and they will be colonized by microbes incredibly well. We will see more microbial cells on the root. We will also see more CO2 consumption, but very interesting to find that even in sorghum, we also were able to observe methane consumption as well. So that sorghum plants were also enhancing methane consumption. So it's immediately become an implementation, which I really like to call as a SAP. It's microbial supplement from desert plant rhizospheres that can maximize greenhouse gas consumption by vegetation. And the goal that we're moving forward and we investigate further, we are looking how we can build this safe ecosystem in which we unite agriculture when we supply minerals, we supply fertilizer, we give sufficient water, plant grows well, but at the same time, soil capacity to consume methane is disturbed. And we know that in native ecosystems, those plants take the capacity to take it. So we're trying to rebuild it by restoring, bringing what we learn from nature all the way to what we will have in to translate it to agriculture. And I believe some of research which Dr. Manish Kumar did in my lab, and I know he will present in a moment, just was to dig deeper and look in the interaction under umbrella, how microbes and plants interact with the concept, how we can use microbes not just to restore capacity to consume methane, enhance capacity to consume methane, but also enhance capacity to consume other greenhouse gases as a source of nitrogen. And this his initial study might lead to some wonderful discoveries in that area. With that, I'm going to switch a little bit gears, and I would really like to present a little bit more about C1 biotech. And methane-consuming bacteria, I just showed you, they're beneficial for plants, but they're also beneficial for animals. And actually, C1 biotech started as a technology to produce animal feed, so known as single-cell protein. It was proven in large-scale production, but now we know we can also use it to produce chemical biofuels and other different things. I'm going to talk about them in a moment. Let me to give you a little bit history of C1 biotech, because it's not just appears right now. The C1 biotech, which was built on methanol, is old as 1980 when the first plant was for single cell production was built in UK. This is Pruton plant. And almost the same, just few years later, the plant which used methane was also built in Russia for production of single cell protein using methanotrophic bacteria. We really have a boom of C1 biotech currently because we have LanzaTech, which comes with its different culture. It's not microbes I discussed today. This is Clostridia anaerobic process. It's syngas fermentation, but it's conversion for chemicals and as well as a single cell protein. There is operational plan constructed by Unibio, and just October last month, Calisio also discussed opening a new single cell protein production in China. So there are different areas, mostly around proteins, but the ability of methanotrophic bacteria go way beyond of that. What we have at San Diego State University here, we have a wonderful team of colleagues, and it's an integrative institute which builds an initiative which unites people with very different expertise from College of Engineering, chemistry, as well as biology, all united with the idea of taking waste as a biogas, not natural gas, biogas as a feedstock to drive biofermentation. And this concept, which biogas will be utilized completely and fully, meaning that it's not only methane which can be converted to renewable fuels, but it also CO2 which can be recaptured because renewable energy. And we put relatively large targets, what we would love to produce. One of the first targets, and I will provide very brief introduction to biodegradable plastic polyhydroxyalkanates, but we're also looking on biomaterials based on proteins. It's basically synthetic silk produced by microbes. So under this initiative, we're moving forward, but what is the core? In my lab, the core of the technology is microbe Metallotuvi microbium alkalophyllum. Why I like this microbe? Because it is incredibly flexible, it is robust, it was isolated from alkaline lakes, it can grow at high pH, easy to keep it in puriculture. It already has high tolerance to carbonate. I will emphasize why it is important. But most likely we have genetic FBA flux balance analysis or everything to build around these microbes to make it a wonderful chassis for biotechnological application. Again, what Dr. Kumar was able to see in my labs a basic pipeline for strain engineering, which starts with cultivation, generation of omics data, and its typical approach. It's design, build, test, and learn approach. We look on the component, transcriptomic, proteomics, gene expression data, do mutagenesis, do phenotyping, integrate everything in computational model, learn out of it, and design further. I want to tell you one project which I personally really like, and this is ongoing collaboration between my lab as well as Johns Hopkins University National Lab. This is uniting electrocatalysis and bioconversion, which I just mentioned to you before. The goal is to optimize electrocatalysis and integrate it immediately with fermentation to produce renewable biobenign plastics. And we have 3 different targets in set. We have to understand baseline parameters for methanol utilization, improve carbon conversion efficiencies. Then we need to integrate PHA producing pathway now microbe because it was a natural producer. And we also wanted to integrate C1 and C2 organic compounds. And I'm taking you away from methane, but it's methane and methanol. Our compounds now all come from electrocatalysis. What I'd like to show you here is that we can fix CO2 and we can efficiently produce currently formate as well as the acetate. Methanol can be produced and methane can be produced, but they're more expensive. However, uniting those different compounds together, we can build polyhydroxyalkanoids at the price as currently are on the market, making the overall technology techno-economically feasible. And that became our task. Can we do it? And we started with establishing pipeline. Microbe is incredibly great at methanol consumption. It has very efficient pathways for utilization. And it also has serine cycle, which drives everything in acetyl-CoA. However, when we run microbe in our robust media, we notice that theoretical parameters quite different from what we see from the culture. And what it basically means that if our theoretical tells us that microbe can produce, should be convert 65% of carbon into biomass, our microbe give experimental only 43. We immediately thought something needs to be improved. So we did several analyses. We looked what carbon counts, what we're losing, and we found that if we will add simple compound to the media like copper, it will stimulate respiration. If we remove copper, we will lose a little bit in the growth, but at the same time, significant portion of the carbon will be assimilated. So it was very easy, straightforward task. We removed media from cultivation varan. Again, microbe grew quite well, and we immediately reached carbon conversion we were looking for. So the next point was, can we integrate polyhydroxyalkanate producing pathway? And I just show you that enzymes were integrated, activities were measured. But when we looked on expected, what we wanted to see polyhydroxybutyrate, we did not see it, but surprisingly we found that microbe can accumulate even better polymer. Instead of polyhydroxybutyrate, which is not ready to use plastic yet, our microbe was able to produce polyhydroxyalkanates known as medium chain polyhydroxyalkanates, and this is already ready to use medical plastic. So the next goal was, can we optimize it further? And this became goal for my student Richard Hamilton. And again, Dr. Manish was able to interact a lot and learn about his study too. So Richard generated a number of different constructs and was able to improve PHA accumulation up to 15%, which is incredibly good for microbe, which use methane or methanol as a feedstock and drives the process out of it. The next part, can we improve everything further and integrate C1 compounds like formate and acetic acid, which much cheaper to produce. And again, similar studies were done, additional modification are introduced. And I'm just showing you, yes, we can do that, and microbe becoming more better at consumption of acetate. Again, taking home messages that C1 substrate are incredible unlimited carbon source. And I just show you from nature in arid ecosystem when they can capture from environment all the way when you can run biotech and produce biodegradable plastic. And of course, by using standard synthetic biology approaches coupled with metabolic engineering, microbes can be improved in their capacity and trained to produce something new. Again, I'm just finishing today with indicating that I told to you only about polyhydroxyalkanates, but over the years, we developed this microbe to produce a number of different compounds, from sugars, enzymes, to other precursors of different polymers. We also built a system in which we can use methanotrophic bacteria to capture methane from not atmosphere, but from the sources where it's about 1 to 5% to completely eliminate it. The technology called SLEEVES. And with that, of course, I will be nothing without my wonderful team. And I emphasize for you today people who contributed to study. I presented Richard Hamilton, who was on metabolic engineering, Nati Delhabre, who worked with Manish Kumar a little bit more. So she is on metagenomic studies as well as a genomic studies. Marcus Brake, who contributed also to understanding of metabolic pathway now, microbe. Of course, Professor Elizabeth Waters, who is a wonderful plant biologist and who provided plants to all studies as well, her insights into plant physiology. And with that, again, it's my great pleasure to be here, and I thank you, and I'm ready to answer your questions. Almost on time.
Thank you so much.
Thank you very much, Dr. Marina, for a nice lecture. In fact, there are 1 or 2 queries. I will not take many. 1 or 2 student research scholars are interested to know. So if you permit, would like to take 1 or 2 queries? Dr. Marina?
Yeah, can you please repeat question?
There are 1 or 2 queries from research scholars who wanted to know something about the research you have done. So they have some questions, not questions basically, they have some queries. So,
Sure. So,
like one of the students is asking about, what about the methylotrophy
oh, it's I
other
look
than
at you.
C1 carbon?
Yes,
If
yes,
you can
I
see the
see
chat
chat.
box. Yes.
Okay, so what about metallotrophy other than C1 carbon compounds? Actually, metallotrophy is based on single carbon compounds such as methane and methanol utilization. However, there are other, and I presented very briefly today, it's LanzaTech microbe, it is Clostridium, which can utilize syngas. It's basically carbon monoxide. It is not organic molecule, it's an organic molecule rich with energy. What is the source of carbon monoxide? We have a number of those sources at steel production. When coal is burned to basically melt steel, there is a flux of CO and hydrogen comes out. It's pollution unless it's eliminated. So LanzaTech unites the technology with a microbe and produce ethanol. And they went even further from ethanol, actually. They produce a jet fuel, and now they're looking on many other aspects as well. It's different technology, it is not really metallotrophy. Now there is new technology which comes out, and I believe it's related to multi-carbon compound and C1 carbon compounds as well. I mentioned to you that electrocatalytically it's incredibly easy and very beneficial, cheap to produce formate. So it's not metallotrophy, it's typically set separately, but many metallotrophs can utilize formate, but same time people separate formatotrophy separately from metallotrophy. It's also area which is investigated quite intensively in Europe right now. Besides formate, acetate can be produced. What we should remember about acetate and formate, even so they're incredibly cheaply to produce from electrocatalysis, from microbial standpoint, they're not really great substrates. So we have to optimize microbes to utilize them. So
Right.
can we say we'll be, yeah, I believe I answered. Please add if I didn't. Now,
Another
can
one,
we say
related one is methylotrophs be a great bioinoculant. Can we say like that? She's tested to know
I
RSI of a
believe
PhD
so.
scholar.
I believe so. With studies which Dr. Manish Kumar did in my lab and more other coming in. I believe one way how I envision, people ask me how I envision methane mitigation. Then people think about filtration system, and I showed it to you. It does not work at the scale. However, when you think about plants' capability to take methane through the leaf and deliver it to the root, for me, it's becoming incredibly easy to implement system for mitigation. So we would love to try it. And I believe that integrating this with the seeds would be a wonderful way to moving forward. However, to do that, we also have to understand in which ecosystem we implement microbes. For example, we now know that in our arid ecosystem, Metallocaldum is almost everywhere. That's microbe which is, unless you convert to agriculture and put tons of fertilizer, Metallocaldum will be in the system. So we believe by re-inoculating seeds, because it will be much easier, we might stimulate community further. But for different soils, it can be investigated. And in some soils, it would be type 2 metallotrophic bacteria, slightly different. And again, I'm kind of giving you something that Dr. Manish Kumar investigated, and he looked on different types and taken the one of the key plants to investigate. But yes, I think it would be good way to moving forward instead of just spreading metallotrophic bacteria around to mitigate methane emissions. I hope I answer your question.
Yeah, thank you very much. Thank you, Dr. Marina. Thank you so much. So, Dr. Manish, I think Dr. Marina is interested to know that what you have learned in last 3 months over there. So it is better to wait. I think you should switch immediately to your experience sharing that what you have learned from last 3 months in the lab of Dr. Marina Kalizinaya, who is actually a great inspiration and guidance for you. And I hope you have learned many new things over there, new equipment you would have handled. So I would like to have your knowledge sharing, you can say, for just 5-10 minutes, you can share your experience with us.
Thank you so much, sir. Thank you so much, Dr. Marina, for your insight into metallotropy and their application in different fields, and particularly C1 biocatalysis and the applications in the plant microbe interaction and the climate change mitigation. And apart from that, I'm going to tell you something or to everyone. So first of all, I'd like to thank everyone, Honorable Vice Chancellor AUMP, and our director, Professor Dr. R.S. Tomar sir, and Chief Coordinator SERB SIRE, Professor A. Chakravarti sir, and my dear students, dear participants, my colleagues, everyone. So I'm going to start with my journey here, whatever so far I have done here. So I think this is visible to all of you. My slide is visible to all of you.
You can make it full screen. Now it's okay. It's
okay.
It's visible now.
Right. So my first day was started with the orientation and biohazard safety, and that was done by Ms. Nathalie Delhare, and she is PhD scholar and teaching associate there, and she explained each and everything related to the biosafety measures and lab hazards there in the lab. And after that, we discussed a lot with Dr. Waters, Dr. Kaliuznia, to execute the project proposal, and then we discussed, we planned accordingly, and everything was discussed in the conference room, and which plant will be taken and why will be taken and how much the seeds will be there and what will be the duration, whether I'm going to complete it within 3-4 months or not. So everything was discussed, and after that we were planned to execute things with the Dr. Waters too. And she's also working well with the plant biotechnology and the evolutionary biology. So the first thing that we initiated, that is related to the hypothesis, that we put the hypothesis that is related to my work. The first one is the methallotropic ability that removed the toxic methane and methanol. The second was the ability to produce plant hormones to stimulate growth, and that is the non-metallotropy. And third one is the consumption of root exudates in the form of carbon substrates and bioconversion. So after that, what we have done, we selected the plant Arabidopsis, and then a number of traits of metallotropes we selected there. And here you can see in the table that we have selected 9 different traits of the metallotropes, and we selected these traits in the form of consortia, because like suppose the first trait, Metallobacteriaceae, that Metallobacterium we have taken, that trait that is working as a consortia, that is working as the combination of microbes of the same group. So different groups of the microbes we have taken there, and 9 different traits were taken to see the interaction of these metallotrophic bacteria in the root rhizosphere of the Arabidopsis plants. So how this was done, this was first selected, the number of traits, 9 traits were selected, then Arabidopsis plants were selected. And because I don't have much time, so I'm going to skip the things very fast. And I know Dr. Marina will be very sleepy after 10 minutes, so I'm going to finish it up very soon, I know. So here in this study, what we study actually, that is very, very important to understand. So first thing is that we are taking here alpha proteobacteria, beta proteobacteria, and the gamma proteobacteria of methylotrophs. And the alpha proteobacteria group of bacteria that we have taken as a trait here, how they are going to interact with the plant root, and beta proteobacteria, how they are going to interact with the plant root, and the gamma proteobacteria, how they are going to interact with the plant root. That is very, very important to understand, and how they are going to mitigate the methane, and how they are going to utilize the different carbon substrate in the root rhizosphere. That was very, very important. So that we are going to see. And here, different alpha proteobacteria like methylobacterium, they are going to utilize methanol, then converting into formaldehyde, then formic acid, then it is going to follow the serine pathway. In the similar way, beta proteobacteria, that is going to utilize methanol, converting formaldehyde, then going to follow the RUMP pathway, ribulose monophosphate pathway, then converting to biomass. And then gamma proteobacteria, it is either going to utilize the pathway of this beta proteobacteria or gamma alpha proteobacteria, and this is also going to convert this into the biomass. So after that, what we have done, we have selected different traits, different bacteria in this study, and the culture purification, the pure culture purification, their identification based on the 16S rRNA gene sequencing was done. So the isolation means not isolation, you can say re-purification was done, and then DNA extraction was done, then 16S RNA gene PCR amplification was done for all of the samples, and they were identified after DNA sequencing. And after DNA sequencing, we got the result, and we were able to identify the traits, whatever the culture, whatever the bacteria of the methylotrophs we had taken as different traits for the inoculation into the plant. So this was all about the bacteria. This was all about the trait that we have taken for the study. And after that, now we are coming to the plant. The plant was selected Arabidopsis, in which we took different seeds of the Arabidopsis and we started to sterilize them. Then after sterilization, vernalization of the plant, seeds were there. And this was the Arabidopsis Columbia type. And this Columbia type strain was taken from Dr. Waters' lab. And we discussed there also with Dr. Waters how to grow them and how much time this strain or this ecotype is taking of this plant Arabidopsis. So this Columbia type, this was taking 7 days for germination. And before inoculation of the bacteria, this was very, very important, that how much amount of the bacterial cells will be there. So that was around 0.2 OD. That was calculated, that was enumerated using the 96 well plate method. And this method was, Dr. Marina taught me this method, how to do this. And I was very happy to learn this technique also. So we were able to do this. And after that, the vernalization, germination was done. Then after 7 to 12 days, we inoculated the different strains of methylotropic bacteria, different traits in the form of consortia that was inoculated into the root of the plants. And then we waited for the plant growth, flowering, and then harvesting. So a number of results are there. So I'm not going to present all results here, all data, all physiological, morphological, or molecular. So many data I have, but still I don't have much time. So I'm going to summarize within a few next minutes. So here we can see in the red circle, you can see these are the controls, and as compared to controls, we have very good results in case of the Arabidopsis plants, where we have seen very good results. And good results were not only in the form of their growth, they were in the form of their the leaf surface area, the total number of leaves, the flowering, and the chlorophyll content, and the rate of photosynthesis, and the root biomass, and everything we analyzed there. And this was very laborious, but we got very good result. In the trial experiment, we didn't get the good result, but in this experiment we got very good result, because we extended the time of inoculation a little bit, and after that we were able to get the uniform result in this case. So different traits, you can see the T5, T6, T7, like that, different traits we were able to get, and these are the results, and the results were very good. And here some photographs we have taken, in which we can see that how they are going to be very, very good as compared to the control. And all photographs were taken in daylight, all means different lights that we are going to expose to the plant for the growth, that were switched off. And in the daylight we have taken the photographs, and we got very good result, and we have seen the good and uniform result of the Arabidopsis plants after the inoculation of the different traits. So here the results, whatever results we have obtained here, the very important thing is that which trait was more responsible, which type of methylotropic bacteria was more responsible, or which type of methylotropy was more responsible for the growth promotion there. So here, this was a little conclusion I have drawn here. You can see in this study, whatever inoculation was there, that was mostly because of the trait or the consortia of alpha proteobacteria, followed by beta proteobacteria, followed by gamma, and then gram-positive actinomycetes. So here, the very interesting thing is that earlier some scientists or some investigation, they reported that Methylobacterium or this Methylobacterium and Bradyrhizobium, they have some very good results. But here, the very good result that we have seen in some of our trait, that is not reported yet. Okay, so here we are planning to go for the bacterial community profiling, their abundance. The soil DNA extraction is already done, 16S metagenome. We are waiting for the 16S metagenome and their sequence results. And so we can analyze accordingly, and then we can see whether there is the actual abundance of our bacteria that is inoculated into the root of the plants or Arabidopsis or not, whether there is the bacterial community profiling and abundance of that particular trait or not. This will be this will be clarified, and this will be, that will be known after this 16S metagenome. And another aspect is what? Another aspect is related to the plant, metabolomics. So now for this, I need to convince, and the hypothesis must be very clear that I'm going to present before Dr. Marina, and then particularly two or three traits or consortia we're going to take for this metabolomic study, in which we can see the classes of different chemicals, those are responsible, and the different metabolic pathway, those are taking place here for the plant growth promotion. And this is, this is the way we are going to mitigate the methane, we are going to mitigate the climate change. And here in this study, the conclusion is that on the basis of this morphological, physiological, molecular characterization and different interaction study, we can say that methylotropes, particularly alpha proteobacteria groups, they seems to have significant potential to enhance the plant growth promotion and enhancing the root colonization ability. And there is the abundance of the community of that particular trait in the form of consortia, and they are going to do the biotransformation or the bioconversion there in the root rhizosphere region. So this is all about my study that I have done. I have so many data, but I am not presenting all those. And here I'm going to acknowledge DST SERB, New Delhi, Amity University, San Diego State University, and Dr. Waters. She helped a lot for plants and the growth of the plants, the inoculation, greenhouse, everything. She helped a lot there. And is Tiago is a PhD research scholar. He also helped there. Then I'm very thankful to Peter, Marcio, obviously Dr. Kaliznia, Richard, and these are another team you can see here, Natalie, Nick, China, Brianna, and Will. Thank you so much, everyone, for your patience.
Thank you, Dr. Manish, for your crisp presentation on your experience in the Dr. Marina's lab. We shall discuss more about it so that we can enter into some collaborative type of the study or research work in Dr. Marina's laboratory. But before that, I would like to request our Honorable Vice Chancellor, Lieutenant V.K. Sharma, USM retired, to share some of the good points that he have captured in this presentation. So as a remark, sir, I would like to request you to please give a valuable remarks on this today's presentation.
So thank you very much, Dr. Marina, for a very insightful talk and good revelation about how even the deserts can be utilized to reduce the biggest greenhouse gases reduction, the methane. So as the report says today, that the world population has doubled since 1974. And now it is believed that after a few years, it will start declining, or it will come down. The bubble is almost at the peak now. That's what the analysis and some of the scientific reports say. But it is a reality today that the global warming is taking place and its effects are being felt all over the world. And it is the duty of all mankind to join together and to ensure that a reduction in global warming takes place so that the mankind is saved from various effects of global warming, which are so evident and which are actually seen happening. We are seeing that the glaciers are melting. We are seeing the change in patterns of the climate, the weather conditions, particularly in our own country, in India, we have noticed that. We have seen the increase in pollution levels in AQI index, we have seen, and all that which is happening. Primarily, there are two gases which are responsible. One is methane and other is carbon dioxide. So these need to be reduced. And the measures which need to be taken, reduction besides scientific research, which Dr. Marina is doing in any case, we need to come out with solutions which are cost-effective solutions and implementable solutions. The two words I would like to emphasize, cost-effective and implementable, and which can be taken at a greater level and soonest. As was done in pandemic times, the drugs were introduced fast without even following the correct route for their verification because there was an emergency. I believe that today also the quality of the air that we breathe, the way that the effect is happening on mankind because of global warming, because of increased greenhouse gases, there is an emergent sort of a situation, and we must come out with ideas to mitigate the reduction and to undertake reduction of these two important gases, that is carbon dioxide and methane. So some of the ways which have been found. Another problem which we are facing here in India is about stubble burning by farmers. It's something which I would like Dr. Manish to see in addition. How is it being handled in the US? How is the stubble being removed after the paddy has been taken out, and what is left? What technology is being utilized? And bring home, that is something in addition to what you are doing, Dr. Manish, that you can look into it if you have time and resources. That is something, a live problem that we are facing in India, especially during this season when the temperatures are falling. So as a result, all these gases, they are at the ground level and they are highly pollutant gases, which are making people, especially in metros in Delhi and in Punjab and Haryana, difficult to breathe. So these are some of the practical problems which we are facing here in India. And I'm sure the other global warming is also taking place, and other countries are equally feeling the pinch of it by various types of torpedoes and all those things which are happening. And so many natural calamities which happen. So thank you very much, Dr. Marina, for giving a very insightful talk, and to Dr. Manish. You are learning, of course, and what is more important is collaboration. Any research must produce new knowledge or increase the existing knowledge. And the best way to happen is through collaborations. And today, the internet has connected the world like never it was before, especially after the pandemic. And with the technology having made available to ensure that we are all connected, I feel that there's a lot of opportunity between Amity University, Madhya Pradesh, through the good offices of Dr. Marina and your university, to integrate and take the research further in various other fields, including the one which we are doing. So thank you very much, Dr. Marina, and thank you, Dr. Manish. Please continue the good work you are doing, and best wishes, Jai Hind.
Thank you,
Sir. Thank you very much, sir. I would like to go further in this regard, with regard to collaborative work. Dr. Manish and Dr. Marina, I would like to request you that in the present context, what is the most demanding one is, I think some session has been given by Honorable Vice Chancellor. It's a good suggestion. We are facing a lot of problem with this stubble burning in India. So can we find some solution for that? And in addition to that, can we have some kind of collaborative research in metabolomics and metagenomics? Because these are latest fields. So Dr. Marina, your viewpoint on this.
Okay, so I believe number one, for agricultural and biotechnologies, I would like to say that even in India, you already have biotech companies which are quite interested on C1 utilization. I was fortunate to talk to CEO of String Bio company who's located in India, and her main interest also, the CEO is also to take methane utilization as a platform, biogas utilization to produce different compounds. So there is already a consortium, and I believe it will be growing and growing. And Manish, it's also kind of wonderful that you contribute to that as well. So that's number one. I think that we will see even more going on in that particular area. Now, regarding to metagenomic and metabolomics, I know that Manish has quite a few of samples, and we would love to keep them and analyze, because when experiments are done, you really want to, you already have collected samples. So basically just submitting them for sequencing or for metabolomic, high throughput metabolomic analysis. What we will discuss with Dr. Manish, and it will happen on Tuesday, we want to sit down and build a very clear hypothesis. What exactly questions we would like to ask, what we would like to get from the experiment, what we already see, and what would be the most reasonable way to analyze data sets, because you can go in a simple way or you can go in very complex way. The experiments Dr. Manish Kumar carried out is multidimensional, and it has so many layers, and some of them can be resolved now, like looking how community interact, which microbe prevail, what microbe was eliminated from interactions, what is the most beneficial in this particular settings. But metabolomics, I know that Dr. Manish Kumar also wants to do it right now as well, but we typically do metabolomics it's really harsh to do metabolomics on soils, because you have a number of different already compounds present in the soil. For our metabolomic experiment before, we did slightly different, and I know that Manish will learn about this technique. We use synthetic sand, which is incredibly pure from any compound. So what we really observe is what is secreted by plants or what is secreted by microbes. We know that there is no interactions other than what was produced there, because everything else was clean. However, it's somewhat artificial environment. So soil metabolomics is incredibly difficult, because you have humic acids, and during the extraction process, it covers signals so much that it's still work in progress and people moving forward. So unless we have a very clear hypothesis about which compound we target and which compound we can look across different settings, it will be like analyzing humic acids, and this is what signal will come out. So we really need to develop this hypothesis. What comes out from Manish Kumar, Dr. Kumar experiment is very unique, that it's alpha proteobacteria, it's very unique serine cycle AMP pathway. That means that methylmalonyl-CoA, ethylmalonyl-CoA plays some role in establishing a collaboration with plants. And it gives us idea, how about we will look on those metabolites, but we definitely have to look on other results as well. And I can give you even more details, but in general, what will be happening next, again, we will analyze data, and I hope we will be able to publish first manuscript. And from that, it's always nice to see not just Arabidopsis, but also looking on other crops, which can be analyzed too. And taking it even further, it would be absolutely incredible to do even field studies. And I know that Manish mentioned this is something he can do. So it's really incredible, just taking one step closer to field, to real world. And I believe just one tool which Dr. Manish Kumar can polish, and this is something you can do with NATI, is just using LGR system to look on methane emissions and kind of know how it also can be done in agricultural settings to collect this wonderful set of information. Dr. Tomar, did I answer all your questions? I missed something. Just please. Okay, good.
Thank you, Dr. Marina. In fact, Dr. Manish might have already got some idea from you, and he might be having some plans in his mind. This was just a suggestion that how we can cooperate further in this regard. Dr. Manish, would you like to say something on this?
I think so much knowledge, the way of working, and the management, and so many things are inculcated from this lab and from Dr. Calizonia I received, and I'm going to execute there also in India, and in future obviously there will be one collaboration with actually I know Dr. Marina since 2012. Am I right, Dr. Marina? Yeah.
I believe so. This is when we have a visitor, Kamarina, before you, and Kamlesh visit my lab in Seattle. Yes, and you just was following up after. Yes, so it was actually wonderful that we have an opportunity to write manuscript together, and I looking forward to a wonderful. The data I saw so far are incredible. We really would love to publish them, and Manish nicely promised me to spend first month when he come back home analyzing data and preparing it for publication.
Right.
And that would be also
Thank you.
incredible because it really builds foundation for the next round of studies.
So,
Yeah.
Dr. Marina, thank
Thanks a
you very
lot.
much for your insightful talks, and we would be looking forward to maybe signing an MOU with between our
SDSU.
university and your university at a later stage.
Right, sir.
Which, we'll work out the modalities and then get back to you. Now I would request Professor A. Chakravarti, who can give his expert views and comments on the interactions and upon the lecture that talk we had from you, and also about the experience of Dr. Manish, which he just shared. So over to Professor Chakravarti.
Thank you.
First, let me tell you, it was an excellent talk. I learned a lot. And I would also like to tell Manish, when Marina was making her presentation, the style of presentation, the way of presentation is also an art which you should learn. What I was seeing, the left-hand side of your presentations with those photographs of the crop fields, their outcome and the output to research was in the right-hand side of the PPT. This is also important, you do high science, but finally you have to communicate that high science. We all know that there is no high technology without high science. And India has a special challenge because we are an agrarian country. But parallelly, we are also obliged in the COP27. So we have to make a balance. India has to feed almost 1.5 billion people. So these are some of the challenges, and that's why this science is important. This particular science is important for India because it brings the balance. While we are committed for high production of crops, but we are also parallelly committed to reduce the carbon emission. This is the balance, and that's why this science, this technology is important. We wanted Manish to spend a few more months extra with you. And I'm sure that Manish, you will explore those opportunities for the future. And Manish, while you were speaking, you took the name of some of the other colleagues of Marina who helped him in deciding I think they are also our important connector.
And Dr. Waters.
Yeah, so I think we would have loved to see them today, but I think they must be occupied with many other things. And Marina, when you were making the presentation, you gave that circle where the biologist, mathematician, chemists are all grouped together. That is the tomorrow's science. That is the tomorrow's technology. Biology connected with mathematics and then both together connected with chemistry will only bring the solutions. The solutions are losing boundaries. Physics, chemistry, mathematics, these are the traditional boundaries are vanishing. So you have to think beyond. And another point I very much liked, Marina, you said Manish did these experiments and generated some very interesting data. Now, how that interesting data will change our line of thinking is the actual brain of a scientist. Data can be generated by anyone, but how to interpret that data, how to use that data for my new knowledge, new innovation. I think, Manish, you are very fortunate that you could have this guidance from Marina and others.
Sure, sir. Without interpretation, we are not going to publish the research paper. So
Yeah,
we are
surely,
in the
surely,
process to
surely.
represent the results.
Surely,
Yes.
surely. And these interpretations actually open up the new path, new avenues to look at the problems, look at the challenges. So we are pretty sure, I think, Manish, this 3.5 months, I think it finished very quickly. We must thank,
Yes, sir.
I must take the opportunity to thank two people for this, rather two. I must thank Government of India for choosing Manish. I must thank Lieutenant General V.K. Sharma in supporting Manish's application. But above all, I would also like to thank the, I think, founder of Amity. You know, I don't know why. Today, you will not believe he's also listening our discussions silently. And he said, I'm not able to come physically in the meeting through the internet, but I'm listening everything. Because he has a great national feelings. It is not only Manish's going, it is also what Manish brings back for this country to make country scientifically advanced and solve problems of 1.5 billion people who are looking at you for such solutions. So thank you, Marina. I'm pretty sure General V.K. Sharma has already told about the possible MOUs, which you may like to consider. But MOUs are no MOU. Partnership should grow. And we would like to see you soon in India.
Thank you. It's wonderful.
And you cannot go to Goa going through Delhi, where we are located.
He is planning for Australia first.
No, we have lost our cricket match in Australia, but that doesn't mean that Australia will be there ahead of us. Now, India is a fascinating country, and Amity, I must tell, it is a fascinating journey which was initiated by our founder president. We are hats off to him, hats off to the leaders like V.K. Sharma, who are carrying this flag flying high and producing our real workforce or real mind force like Dr. Manish Kumar. So Manish Kumar, it is not only a 3.5 months holiday journey. It was a trip which you must cherish throughout your life, and it must begin to show what we can do when we join hands. So over to you, Dr. Tobar, and thank you once again, Dr. Marina, to be here with us.
Thank you very much, sir, for your nice words about today's this particular program. Basically, a guest lecture by Dr. Marina, is nicely presented and given a lot of insights in the new fields that are coming up. And very soon we shall be entering into some MOU, as suggested by our Honorable Vice Chancellor. So, ma'am, I request you be ready for that.
When the manuscript is coming up?
Very soon.
Good, you're almost like me. By the end of the year, that's Manish Kumar present for me for New Year.
Yes, certainly. Yes. We will seek your input as well on the appraisal.
Yeah, thanks a lot. Thank you very much. So now, before I close this program, today's program, I would like to request Dr. Isa Sood of our FSTIA to give vote of thanks today. Thank you very much.
Thank you so much, sir. I, on behalf of Amity Education Group, deem it a great honor and privilege to propose the vote of thanks for today's event. First and foremost, I thank our special guest of today's event, Dr. Marina. We are pleased to have your valuable presence and listen to you today. Your inspirational words have enlightened us and gave us the insights about the Earth's climate, plant microbes, importance of methane as an organic compound on Earth, and its role in global climate. We now aspire how we continue our partnership with you as your mentorship. This important research is of great importance, not only to India and US, but for the entire humanity for its food safety. I'm obliged to our Honorable Founder President, Sir, Dr. Ashok K. Chauhan, for his great vision, encouragement, and valuable direction. He only inspired us for availing this fellowship. Not only this, he goes beyond it. We must exploit this opportunity. This is his vision. Today's event is actually incited by him. Sir is an inspiration for all of us, and we always seek his blessings. Dr. Manish, you have added a feather to Amity's hat, and we are always proud of you. We always wish for your great success. You are fortunate that you have been chosen by Government of India for this fellowship, and you are representing India there. It's just a beginning for you, and you have to take this scientific journey further. I express my heartfelt gratitude to respected Vice Chancellor Sir, Lieutenant General V.K. Sharma Sir, and his entire team for his guidance. Sir, this is all because of your never-ending guidance, encouragement, and motivation to the faculty members and students that they are able to grow and avail such opportunities. I would also like to thank all our participants, including faculty members, researchers, and students, for their patient listening and wholehearted participation. I'm sure there are many takeaways for them for this session. I would like to thank Professor Chakravarti for his mentorship, guidance, and support to not only Dr. Manish, but to all the SERB fellows who are availing this fellowship at various countries abroad. This event would not be a great success without your involvement, sir. A special thanks to our IT team for the smooth conduct of the event. Vote of thanks is incomplete without thanking the entire backend team involved in SERB, especially Ms. Shefali Kashyap and Ms. Kamakshi. At the end, I'll say, coming together is a beginning, staying together is a progress, and working together is a success. So through this webinar, we have taken the first step of coming together to work together. Thank you all for making this event a success.
Thank you.
Thank you so much.
Thank you
everyone.
so
Thank you.
much.
Thank you,
Thank
Ms.
you so
Isha,
much.
for your
Thank
nice
you.
vote of thanks. Thank you very much. Thank you all.
Thank you, sir.
Namaskar. Thank you.
Thank you all.
We can close.
Okay, we can close the meeting.
Yeah, we can close the meeting now. Thank
My
you.
pleasure to see you. Thank you, everyone.
Thank you.
Thank you so much.
Thank you.
Bye-bye.